Hi there, and welcome to episode 1 of the MCAT Prep Pod Biology Review. I'm your host Mark, and we'll be doing quite a lot of learning and reviewing over this series in order to help you do your best on the MCAT. I originally wrote and hosted this podcast way back in 2016 just as a way to help me learn during my workout time or my cleaning time. But between new school program and my other family obligations, I had to shut down my hosting server since I just didn't have enough time to maintain it. But I'm back, baby, and hopefully for good this time. And I've been going through and updating and remastering all my old recordings so they might help you with your studies now. I've been working on new recordings, re-recording episodes that had poor audio quality, making sure that all the content is there and up to date, and filling out all the rest of the MCAT tests. So please drop by my website, reviewmcat.com, and check out what we have available now. If you join the mailing list there, I'll let you know when new episodes drop, and you can access the written notes that go along with each episode. The written notes are great. They have tons of informative figures, and also they're kind of a nice compressed version of everything that you need to know. So anyways, I know you're here to review, so let's get to it. In this episode, we're going to cover some of the biology basics. We're going to talk about cell theory, eukaryotic cells, and a brief coverage of tissues. We're going to start small, and then we're going to build up to more complicated as we go. So that way we can build some momentum, a little bit of confidence, kind of get rid of the easy pickings first. So without further ado, let's get right into it. Starting out with cells here, in 1665, Robert Hooke first observed cells, paving the way for the rest of cell theory. And the core tenets of cell theory are that all living things are composed of one or more cells. The cell is the basic structural and functional unit of life, and all cells come from pre-existing cells. Cells also contain genetic information in the form of DNA, which is passed from parent to daughter cells. Cells in general can be split into two basic cell types. Those are eukaryotic cells and prokaryotic cells. Eukaryotes would be those with a cell nucleus, like your plant cells or your animal cells, and prokaryotic cells would be those without a nucleus, like your bacteria and your archaea. In this episode, we're going to mostly focus on eukaryotic cells and their organelles, and the eukaryotic cell is a collection of organelles all held together by an outer semi-permeable double-layered lipid membrane called the plasma membrane. Inside the eukaryotic cells, you have membrane-bound organelles, each with specific functions. First, let's talk about the plasma membrane a little bit. This is the boundary between the cell's internal environment, or the cytoplasm, and the external environment. It acts as a dynamic, selectively permeable structure that's crucial for maintaining cellular integrity, controlling transport, and facilitating communication. The currently accepted model for the membrane structure is called the fluid mosaic model. So breaking that down, first, the membrane is fluid. It's not rigid, so it consists primarily of a phospholipid bilayer. And if you remember phospholipids, they have a hydrophilic, water-loving phosphate head, and they have two hydrophobic, water-hating fatty acid tails. So they'll arrange themselves spontaneously in water with this tails facing inwards or towards each other away from the water, and the heads facing outward towards the aqueous environment, either inside or outside the cell. This bilayer forms a fluid matrix where individual phospholipids and proteins can kind of move around on the membrane. The fluidity is influenced by temperature, so a higher temperature means it's going to be more fluid. The type of fatty acids that are in the membrane also influence the fluidity, so more unsaturated fatty acids increase fluidity, and more saturated fatty acids decrease fluidity. And also the presence or absence of cholesterol also affects the fluidity. More cholesterol makes the membrane less fluid. So if you think about how butter, which is saturated fats, is a solid at room temperature, that's because the molecules can pack tighter, they can interact with each other a little bit more, versus something like olive oil, which is less saturated, and at room temperature that's a liquid. So saturated fats are going to be less fluid, and unsaturated fats are going to be more fluid. The mosaic part of the fluid mosaic model means that embedded within or attached to this lipid bilayer are proteins, and these proteins form a mosaic pattern. There are integral proteins that penetrate the hydrophobic core of this membrane. They're often spanning the entire membrane. These are called transmembrane proteins. They can function as channels or carriers, pumps, or receptors, where if they sense a change on one side of the membrane, they can change their shape on the other side of the membrane to essentially signal through the membrane. There are peripheral proteins that are loosely bound to the surface of the membrane. Those are often integral proteins or lipids, and those can act as enzymes or be involved in structural support or signaling for the cell. Attached to these proteins on the outside of the cell, there's very often short carbohydrate chains. These are forming what are called glycoproteins, and there can also be these carbohydrate chains attached to the lipids on the outside of the cell as well, and those are called glycolipids. These are exclusively on the outer surface of the plasma membrane, and these carbohydrates that are attached, they form a layer called the glycocalyx, and this plays an important role in cell-to-cell recognition. It's involved in adhesion, and it's involved in cell-to-cell signaling. The plasma membrane is also selectively permeable. Due to the hydrophobic interior of the lipid bilayer, small nonpolar molecules like O2 or CO2 can actually pass pretty easily through just with simple diffusion. But if you look at small polar molecules like water, it's a little bit harder for things like water to pass through there. It can pass, but it's a lot more slowly. So there are special channels, things like aquaporins, to facilitate things like water transport. And ions like sodium, potassium, and chlorine, if you remember, those are charged particles. So sodium and potassium have a positive charge, chlorine has a negative charge. Those will not pass through the lipid bilayer, at least not on their own. They require some kind of transport protein to get through the lipid bilayer. This is also the same case for the larger polar molecules like glucose or amino acids. So those also can't pass through the membrane. They need some kind of way to get in. Maybe it's a protein or some kind of transport. So some of the ways in which cells transport things across the membrane are passive transport. And passive, the word passive means it does not require cellular energy, or ATP. So this is movement either down a gradient or down an electrochemical gradient or down a concentration gradient, where you have high concentration of this thing on one side of the membrane and low concentration on the other side of the membrane, and these move from high concentration to low concentration. And so these can be simple diffusion, like I mentioned, movement of small nonpolar solutes that are going directly through the bilayer, like oxygen and CO2. So maybe the cell's building up CO2, it's in use, and there's not a lot of CO2 on the outside of the cell, so the CO2 can just leave the cell freely. And on the outside of the cell, maybe there's a lot of oxygen, and the cell's low on oxygen, so the oxygen can just freely diffuse across the membrane. Things also can passively go through the membrane through osmosis, and this is the specific diffusion of water across a selectively permeable membrane. And this is from an area usually of low salt or solute concentration to an area of high salt or solute concentration. So on the topic of osmosis, I'm going to briefly cover the effect of the surrounding solution on water movement called tonicity. And you'll probably remember this from most basic biology courses. If you put a cell in an isotonic solution, that's where the concentration of solutes inside the water or the solution is the same as the concentration of the solutes inside the cell. There's no water exchange, there's no ion exchange going on through the membrane, and the cell will be happy. It won't change its size, there's no water movement. But if you put it inside of a hypertonic solution, that's where the solute concentration outside the cell is higher than the concentration inside. Water is going to move out of the cell by osmosis, and this is going to cause the cell to shrink. And finally, if it's moved into a hypotonic solution, that's where the solute concentration outside the cell is lower than the concentration inside. water is going to move into the cell by osmosis, and this is going to cause the cell to swell up, and it could even potentially burst or lyse. So understanding tonicity is crucial for predicting how things like red blood cells are going to behave in different environments. And the last method of passive transport I'll talk about here is called facilitated diffusion. And this is movement of ions or larger polar molecules down their gradient with the help of membrane proteins. So this is either a channel protein, which can form pores, or a carrier protein, which can change shape when something binds to it. And this is still passive. There's no ATP or energy needed for this. Like I mentioned, some of the ions like sodium and potassium can't cross the membrane on their own, so these guys might need a channel to open so they can go through the channel. So that's facilitated diffusion. This brings us to active transport, and this is the second way in which cells transport things across the membrane. And this requires cellular energy, which is usually done through ATP hydrolysis. So the cell has to use ATP to move these substances against their concentration or electrochemical gradient. So this is from low concentration or low potential to high concentration or high potential. So imagine moving something up a hill. It doesn't want to just go down. You have to push it up with energy. There are two types of active transport I'm going to talk about here. There is primary active transport and secondary active transport. Primary active transport uses ATP directly to power the transport. The classic example here is the sodium-potassium pump, where there's a sodium-potassium ATPase. This pump selectively transports 3 sodium ions out of the cell and 2 potassium ions into the cell for every ATP molecule that's hydrolyzed. This maintains the electrochemical gradients across the membranes necessary for things like nerve impulses and a lot of other crucial functions. We're going to talk about this in detail in the nervous system section, so I'm not really going to go into the details of this now, but that's an example of primary active transport. There's also secondary active transport, or also known as co-transport. And this uses the energy stored in electrochemical gradient, which is usually first established by primary active transport, to drive the movement of another substance against its gradient. So this can be something called symport, where both substances move in the same direction, or antiport, where two substances move in the opposite direction. But the key here is that one thing is moving against its gradient, and that's the thing being transported, while the other thing moves with its gradient. So one thing going with its gradient, one thing going against its gradient, and it's mediated by proteins at the membrane. The last form of transport here I'm going to talk about is bulk transport, and this is for moving large particles or large volumes of substances across the membrane, and this involves vesicle formation and requires energy. So there's endocytosis, which is bringing material into the cell by forming vesicles from the plasma membrane. And so this is like phagocytosis, which is the cell eating or engulfing large particles. This can also be pinocytosis, which you can think of as like the cell drinking, or it's taking in the extracellular fluid. And this also can be receptor-mediated endocytosis, which is the specific uptake which is triggered by a ligand binding to a receptor. So the cell wants to take something in, it binds it with a receptor, and then it does endocytosis and brings whatever that is into the cell. There's also exocytosis, and this is releasing material out of the cell by fusion of the vesicles with the plasma membrane. So when a cell wants to secrete hormones or neurotransmitters, it will essentially first make them, and they'll be packaged into vesicles. They'll process through the Golgi apparatus, and then they'll fuse with the cellular membrane, and those things will be expelled out of the cell into the external environment. And there's actually a really nice video that I suggest you watch. It's called, I think, Inside the Cell. I'll have it linked in the show notes, and it's a really nice 3D animation of some of these processes going on inside the cell. They show exocytosis. They show a lot of other good things in there, so it's really worth watching. It only takes a few minutes, and it'll give you a good idea of what's going on visually inside of cells when some of these processes happen. So finishing up the plasma membrane section, plasma membranes have receptors, which I have mentioned before. And these are integral proteins that bind to specific signaling molecules, or also you could call them ligands, on the outside of the cell, and these trigger a response inside the cell. This is fundamental to cell communication, and we're going to discuss this a lot more later on in the series. So the plasma membrane, it's this dynamic, complex structure. It's essential for maintaining the cell's internal environment and mediating its interactions with the outside world through this carefully regulated transport across the cell. Okay, now we're going to get to some of the organelles that are contained inside the cell. The first one I'm going to talk about here is the nucleus, which contains the cell's genetic material, or the DNA, and it's organized into chromosomes. This is enclosed by a double membrane, which is called the nuclear envelope, and this double membrane has nuclear pores that regulate transport in and out. The DNA inside the nucleus exists as chromatin, and there are less condensed euchromatin, which is generally active DNA that's undergoing transcription, and there's highly condensed heterochromatin, which is generally inactive DNA that's packaged around histones, and it's not needed for that cell, or it's not needed at that time. So if you look from one cell to another in an animal, the euchromatin and heterochromatin would likely be different depending on the cell type, since certain cell types will need a certain set of DNA, while other cell types will not need that DNA, but they'll need other DNA. Inside the nucleus, there is what's called the nucleolus, and this is the primary site of ribosomal RNA synthesis and the ribosomal subunit assembly. And the ribosome is something that translates RNA into DNA, so this is an important part of your cell here. The next organelle we're going to talk about is the mitochondrion, or plural is mitochondria, and this is typically called the powerhouse of the cell. It has two membranes to it. It has a smooth outer membrane, and it has a highly folded inner membrane forming what are called cristae at the folds. The space inside the inner membrane is called the mitochondrial matrix, and this is the site of the Krebs cycle, which we'll talk about in the biochemistry section. And the space between the membranes is called the intermembrane space, and this is where the proton gradient is established during electron transport, which we're also going to go over quite thoroughly. Mitochondria are the main sites of aerobic respiration, electron transport chain, and oxidative phosphorylation that all occurs in the inner membrane, and those are the ATP generating processes in your cell. The mitochondria have their own set of circular DNA and ribosomes, and they replicate all on their own inside your cell. And so this supports the endosymbiotic theory for their origin, which it suggests that early on in life, at some point, the mitochondria was its own prokaryotic organism, and this organism was engulfed or eaten by an early animal cell. And rather than being actually digested by this animal cell, it continued to live, and it produced energy for the animal cell, and it got food from the animal cell. And so it was kind of a mutually beneficial thing for both cells. So the mitochondria just lived inside the cell, and the cell was better off for it, and so they both kind of thrived in this environment. And that is the endosymbiotic theory for their origin. All the mitochondria in your body are inherited from your mother. That means they are maternally inherited, and they also play a role in initiating apoptosis. And interestingly, they're present in both sperm and they're present in eggs. There's actually a lot of them in sperm, but after fertilization, only the mitochondria that are present in the egg actually stay alive to regenerate in the newly fertilized egg. The next organelle we're going to cover here is the lysosomes, and these are membrane-bound sacs that contain hydrolytic enzymes that function around acidic pH at around 5. These are used to break down waste materials, break down cellular debris. They're involved in autophagy, and they're used to break down other ingested pathogens. These can release enzymes that can aid with protein breakdown during apoptosis. And so lysozymes, to lyse something is to break it or to take it apart. And so you can think about lysozymes, they're involved in breaking things down. We're going to move on to the endoplasmic reticulum, or the ER. And this is a network of membranes that are continuous with the nuclear envelope. So they're kind of attached, continuous with the nuclear envelope. And this ER is divided into both the rough ER and the smooth ER. And the rough ER is the part of the ER that has a lot of ribosomes that are attached to it. It's the site of synthesis for proteins that are destined for either secretion or insertion into a membrane or delivery into certain organelles like the lysosome, where the environment inside the organelle is more like the environment outside the cell. It's also involved in protein folding and modification, like glycosylation or other post-translational modifications that proteins can get. The other part of the ER is the smooth ER, and this is the ER part that does not have ribosomes attached to it. And its functions include lipid synthesis, including steroids like cholesterol, detoxification of drugs and poisons. So the ER is actually very prominent if you look at liver cells. They have a huge ER because liver is what's detoxifying your blood. And the smooth ER is also involved in calcium storage, especially in muscle cells where it's called the sarcoplasmic reticulum. There is the Golgi apparatus, and this consists of flattened membrane sacs called cisternae, and the Golgi modifies, sorts, and packs proteins and lipids received from the ER for secretion or delivery into other organelles. It's kind of like the packaging center for the cell. Things enter on the cis face and they exit on the trans face, and the cis face is the face that's facing the inside of the cell, and the trans is the face facing the outside of the cell. This is also involved in post-translational modifications, formations of lysosomes, and the environment inside the Golgi is more like the environment that is actually outside the cell. There are peroxisomes, and these are organelles that contain enzymes like catalase to break down things like hydrogen peroxide or H2O2. H2O2 is a toxic byproduct of metabolism, and it's also involved in beta oxidation of very long-chain fatty acids and synthesis of certain phospholipids from the pentose phosphate pathway. Let's talk about the cytoskeleton, which is the cell's internal scaffolding. This is essentially the cell's internal skeleton, and it's also kind of like a highway system. It's a dynamic network of protein fibers that are running all throughout the cytoplasm of eukaryotic cells. Its crucial functions are providing structural support to maintain the cell shape, enabling the cell to move, facilitating the transport of organelles and vesicles within the cell, and it anchors the organelles all in place. It's composed of three main types of protein filaments. Those are microfilaments, microtubules, and intermediate filaments. To go into a little more detail in some of these microfilaments, these are actin filaments. They're the thinnest filaments. Like if you look at the name microfilament, very tiny filament, they're composed of solid rods formed by the polymerization of actin protein monomers. And it's G-actin polymerizing into F-actin. And polymerizing just means kind of stacking together and coming together. They exhibit polarity, so they have a positive end and a negative end. So if you think about like Legos, you could only stack Legos in one direction. One side is different than the other, so they only stack one way. And this is crucial for their assembly and their interaction with other motor proteins like myosin that move along these microfilaments. They have key roles in muscle contraction, which is an actin-myosin interaction. They have key roles in cell motility. So if you can imagine some cells like an amoeba that move around kind of without, just by like rolling in a way. And they're also involved in the formation of the cleavage furrow during cell division in animal cells and maintaining the cell's shape. And so that's microfilaments. Microtubules are the thickest cytoskeletal components, and these are hollow tubes that are constructed from dimers of alpha tubulin and beta tubulin in like a ring. So if you have the show notes, I've got a picture of all these different filaments here, and the microtubules, you can see how they're formed. It's a ring of these proteins, and like microfilaments, they have a polarity. They have positive and a negative end, so they only stack and go in one direction. And this helps direct movement of motor proteins like kinesin typically moves towards the plus end, dynin typically moves towards the minus end, and again, I would say check out the video that I mentioned earlier. I think it's called Inside the Cell, and I'll have this linked. You can see kinesin and dynein moving on these microtubules. It's a really cool example. And the microtubules, they have key roles in maintaining the cell shape, resisting compression, forming the core of cilia and flagella for motility, which we'll talk about in a minute. They have key roles in chromosomal separation during mitosis and meiosis. They form the mitotic spindle, and they serve as tracks for intracellular transport of organelles and vesicles via these motor proteins I mentioned. They have dynamic instability, which means they can very rapidly grow and polymerize, and they can very rapidly shrink and depolymerize. And this is often driven by GTP hydrolysis associated with the tubulin dimers. And this allows the cytoskeleton to very quickly remodel itself. The last type of filament that I mentioned was an intermediate filament. These are intermediate in diameter between microfilaments and microtubules. They're a diverse group. They're made of various proteins like keratin. They're in your epithelial cells, hair, nails, vimentin, desmin in muscle cells, and nuclear lamins, which support the nuclear envelope. These have key roles primarily to provide tensile strength to withstand mechanical stress and maintain the cell shape. They're very important in anchoring the nucleus and other organelles, and they're involved in cell-to-cell adhesion, for example, desmosomes. And these intermediate filaments, unlike the microfilaments and microtubules, they're generally more of a permanent stick, and they're a lot less dynamic. The last thing we're going to talk about inside the cell is the centrosome and centriole. The centrosome is the primary microtubule organizing center inside of animal cells. It's located near the nucleus, and it has two centrioles that are oriented perpendicular to each other. So each centriole is kind of a cylindrical structure, and it has 9 triplets of microtubules arranged in a 9+0 pattern. And so that essentially means there are 9 triplets all in a ring. And if you have the show notes, I have some of these examples of the 9+0, and there's a 9+2 structure we'll talk about in a minute here. That's essentially if you were to cut one of these centrioles in half and look at the end of it, you'll see 9 triplets in a ring. During cell division, the centrosome duplicates itself, and the two centrosomes, they migrate to opposite poles of the cell, and they organize the microtubules in the mitotic spindle, and these are essential for separating the chromosomes. One thing to note is that plants and most fungi actually do not have centrioles, but they are still able to effectively organize their mitotic spindles. Now moving kind of towards the outside of the cell, there are cilia and flagella, and these are the hair-like kind of whip appendages that are involved in movement. So cilia are typically shorter and more numerous. They're usually involved in things like moving fluid over the cell surface. There's a lot of them in your respiratory tract, and in there they work to move things outside of your respiratory tract to get, maybe you have some dirt particles that come in, and they are working to move things across the cell surface. Flagella, on the other hand, are usually long, and there are fewer of them. Oftentimes there's just one, like for example in sperm, it has a single flagella that's at the end, and that propels the cell through a fluid. These have the 9+2 structure that I had mentioned. If you cut one of them in half, you would see 9 doublets of microtubules arranged in an outer ring, and then you would see 2 central microtubules in the middle. Their movement is generated by the motor protein dynein, which uses ATP to kind of walk along one microtubule doublet, and this causes it to slide relative to the adjacent doublet. So it's kind of like a compressing one side, and that will bend the flagella in one way. If you do this enough, you can kind of get a whip-like motion going around, and then you can cause the flagella to move, and you can move the cell. So now that we've covered cells, I'm going to briefly touch on tissues. Tissues are groups of cells that are organized into tissues. These can be further organized into organs, but we're not quite ready to talk about organs yet. The types of tissues that we're going to cover in the series are epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Epithelial tissue is the most common tissue. It covers your body's surfaces, it lines cavities, organs, it's involved in protection, secretion, absorption, filtration, and sensation. It forms the parenchyma, which is the functional part of most organs. Most cells in your body are typically polarized, which means they have a distinct apical and basal surface. So one side of the cell is different than the other side of the cell, and they rest on these basement membranes. If you imagine, most cells have some kind of function to do something on one side a lot of times and something else on the other. For example, cells on your stomach, one side is going to be facing the stomach environment and the other side might be facing some blood supply environment, and so they're going to be polarized. The connective tissue is exactly what it sounds like. It supports and connects and separates different types of tissues and organs. It forms what's called the stroma, or the support structure. It's characterized by cells like fibroblasts, chondrocytes, osteocytes, adipocytes. These are all different types of tissue that we will talk about. Osteocytes, those are your bones. Adipocytes, those are your fat cells. And so we'll get to these later on in the series. They're embedded in an extensive extracellular matrix that's composed of substances like fibers, collagen, elastin, and some examples of these that I had kind of mentioned are bones, cartilage, blood, adipose tissue, ligaments, tendons. We're not really going to talk too much about muscle and nervous tissue now because we're going to go into those in a lot more detail later, but they're worth mentioning. They're just tissue types here. And your tissues can be classified by a couple ways. First off, they're layers. You can have a simple tissue, which is one single layer of cells, and all cells touch the basement membrane. You can have a stratified tissue. That's where there are multiple layers, and there's only the bottom layer touches the basement membrane. The upper layers provide protection. There are pseudostratified tissues, and those are where they look like multiple layers due to the nuclei being at different heights, but all the cells actually touch the basement membrane, and these are often ciliated, especially in the respiratory tract. And then you can also classify tissues by their shape. So there are squamous, which are flat scale-like cells. Those are thinner. They're usually more involved in diffusion. There are cuboidal cells, which are cube-shaped, which are equally tall and wide. They're good for secretion and absorption. And there are columnar, which are tall and column-shaped. And these are often involved in things like absorption or secretion, like in your digestive tract. So you could call a certain type of tissue simple cuboidal. That would be a single layer of cells, and they're all cube-shaped. So that's everything I wanted to cover for this episode. I know it's all pretty basic stuff, but we're definitely going to get much more complicated very quickly, and this is all fair game for the MCAT. So we'll just do a quick recap on today's episode. We talked about the foundations of cell theory, which are all living things are made of cells. Cells are the basic unit of life, and all cells come from other cells. They carry genetic information as DNA. We talked about eukaryotic cell structure, focusing on the plasma membrane, a selectively permeable lipid bilayer described by the fluid mosaic model. We talked about passive transport like diffusion and osmosis, active transport including the sodium-potassium pump, and bulk transport like endocytosis and exocytosis. We talked about the nucleus, where the DNA is held. We talked about the mitochondria, the site of ATP production, and its role in apoptosis. We talked about lysosomes for kind of the cleanup crew, the endoplasmic reticulum, where you have the rough ER for protein synthesis, and the smooth ER for detox and other things. The Golgi apparatus, which is kind of the packing center of the cell, the peroxisomes, which help get rid of those free radicals from your metabolism, like H2O2. And we talked about how these all tie together in the endomembrane system. We also covered the cytoskeleton, microfilaments, those are the smallest, the thinnest actin filaments, microtubules, which are the tubes, and intermediate filaments, which are essentially the proteins and all their roles in structure, transport, and cell division. We talked about the cilia and flagella powered by dynin, the 9+2 structure that are in cilia and flagella. And remember, we also talked about the centriole, which has the 9 plus 0 structure, that's 9 triplets. And we finally finish with tissue types. There's epithelial tissue for lining and protection. There's connective tissue for support and structure. And then we talked about muscle and nervous tissue. We also looked at the tissue layers: simple, stratified, pseudostratified, and squamous, cuboidal, and columnar. And I think you should really look at a picture for these. I have a picture in the show notes for the tissue classifications. It's one of those things where looking at a picture is probably a little bit better than explaining it. And that's basically everything. This should give us a solid base to build on, and like I said, things are going to get pretty complicated pretty fast here. Thanks for hanging in there with me, and I would love to hear from you. If you have anything you wanted to mention or ask or comments or anything, please feel free to reach out. mark@reviewmcat.com. Like I said, come by the website. We'd love to hear something from you. If you have a problem with the show, or I said something wrong, or there's any kind of error there, please reach out. I'm happy to correct anything that's wrong on the record. I try really hard to make everything right and do my pronunciations of things right, but of course there's always going to be some kind of error there. So reach out. Like I said, I'd love to hear from you. And then the other thing you can do is tell your friends about us, and wherever you found this podcast, if you could leave us a 5-star review, that would be amazing. So hang in there. Great job studying with you today, and I look forward to seeing you guys in another episode. Ta-ta for now.